Hi everyone. Can you see the slides?
Yeah,
can
I can
you
see
hear
the
me?
slides here and see you. That's good.
All right. Well, thanks so much for being here. I would like to present the results of the project in which my colleagues and I examined processing of words and pseudowords in the thalamus and the subthalamic nucleus during a reading aloud task. So reading words out loud depends on our ability to transform information from print into meaningful speech sounds. And this can be achieved through a series of linguistic operations like the extraction of visual features and graphemes from the written form, accessing and retrieval of lexical, orthographic, and phonological representations, and accurate phonetic articulatory encoding. While words can be read aloud following this lexical route or the lexical semantic route, new words or pseudowords have to bypass this lexical semantic level because they do not have corresponding representations in the mental lexicon. And they have to rely more heavily on the grapheme to phoneme conversion processes. So this schematic shows that for words, the decoding via the lexical route takes faster compared to the grapheme to phoneme conversion. However, when a pseudoword like door is processed, automatic lexical search continues for longer, but to no avail. And meanwhile, all graphemes are converted to phonemes via this non-lexical route. Indeed, functional neuroimaging studies demonstrate that pseudowords elicit greater activation in the inferior frontal gyrus, specifically the BA44 area, presumably due to the grapheme to phoneme conversion processes, and BA45 area, presumably due to the hampered lexical search. So apart from genuinely linguistic computations, word processing relies on the scaffolding from what is usually considered executive functions, such as competitor inhibition, memory updating, initiation, sequencing, motor control, self-monitoring, error correction, and processes like that. So the subcortical nuclei, which you see here, the thalamus and the basal ganglia, have been recently proposed to play a role in language processing, having widespread connections with the cortical areas through the cortico-striatal thalamic networks. They are well positioned to serve the cognitive pattern as cognitive pattern generators or relay stations, which may control excitability of the cortical regions and organize neural activity underlying various aspects of cognition through assistive processes, such as the ones that I just mentioned above. So we were presented with a unique opportunity to record directly from the subcortical nuclei from the thalamus in patients with essential tremor, specifically the ventral intermediate nucleus of the thalamus and the subthalamic nucleus in patients with Parkinson's disease. The questions we were after in this study is whether the STN and the thalamus participate in language processing, specifically lexical processing, and if yes, what are their contributions and how do they compare? So we had 11 patients with the essential tremor and 13 patients with Parkinson's disease perform a reading aloud task in which we had 120 words, like fate, they were all monosyllabic, and the corresponding number, a corresponding number of pseudowords like fape. And they were balanced on some psycholinguistic parameters like frequency, bigram, frequency, phonological neighborhood, etc. Okay, so I will go over the results for each patient group. So speaking of the essential tremor patients data, behaviorally, they show differences between words and pseudowords in reading latencies. Their neural data came from 117 recording locations from both the left and the right VIM of the thalamus, which is depicted here in this orange color, and the right and left contexts are superimposed in the same plane. Local field potentials data were transformed using wavelet transfer decomposition into time frequency domain in which the subsequent analysis were performed. So in this grand average neural activity plot, you can see that there is an increase in power in the high gamma frequency range from 70 Hertz and upwards, and a decrease in power in the beta frequency range from 12 to 30 Hertz. Of note is that high gamma increases at speech, the spoken response onset, marked with this dashed line here, and beta decreases at the stimulus onset presentation, which is marked with this dashed line here. Further time logging analysis confirmed that indeed most significant decreases in the beta band marked with these red asterisks here are time logged to the stimulus presentation on the screen, either word or a pseudoword, while high gamma responses increase, most increases in the high gamma responses are time logged to the speech onset of the spoken response, which is represented with this bold black line. Overall, we had 91 significant electrodes with the contacts with significant high gamma increases and 66 contacts with significant beta decreases, which were included in the subsequent analysis of lexicality. So data were averaged for each frequency band, high gamma and beta, and each hemisphere, the left VIEM and right VIEM of the thalamus, separately for words presented in blue and pseudowords in red. So what we saw is are significant differences between words and pseudowords in both the left and the right VIEM of the thalamus. And these differences started to emerge around the onset of the presentation of the word or a pseudoword in the screen, marked with the time 0 in these plots right here. For the gamma band, significant differences were observed only in the left hemisphere, right after the spoken response onset, which is marked with the zeros right here. Now, so looking at Parkinson's patients, they showed behavioral differences in proportion of error in reading latencies between words and pseudowords with higher values for pseudowords compared to words. Their data came from 91 recording locations only in the left STN. We did not have any data in the right STN. So you can see a similar plot here with grand average neural activity. And we also see significant increases in power in high frequency bands and decrease in the beta power. However, you see that most of the increases are concentrated in the low gamma frequency range, not the high gamma frequency range, as in the thalamic data. And beta decreases are concentrated around spoken response onset, like in the thalamus. We did not observe a significant effect of lexicality in the subthalamic nucleus like we did in the thalamus. However, when we subtracted words trials from pseudowords trials, we observed that there is an overall greater low gamma activity, which was not aligned with either the stimulus onset, you see this dashed line here, or the response onset. Rather, it appeared with a temporal jitter across the whole relevant epoch. So to summarize, so our results are the first to demonstrate significant modulations of thalamic and subthalamic LFP neural activity during reading aloud, suggesting their involvement in language processing. Thalamic high gamma and STN gamma and high gamma modulations primarily occur in the spoken response time window, not the time window when participants see the word or the pseudoword on the screen, which indicates involvement in processes associated with phonetic and prokinetic articulatory mechanisms. Increases of activity in high frequency bands is very often accompanied with decreases in the alpha and beta frequency bands in the literature. So we interpret this stimulus-related beta decreases as a signal of change in the sensory motor and cognitive states in preparation for the execution of voluntary movements like speech. So the second notable finding concerns the way in which the thalamus and the subthalamic nucleus encode lexical distinctions, pointing at their distinct functions in lexical processing. So in the thalamus, we observed bilateral stimulus-aligned lexical effects in the beta band and speech-aligned left lateralized lexicality effects in the high gamma band. And the time course and the pattern of this activity is very similar to what has been observed in the cortical regions. So this suggests that regionalization of high gamma activity for language function in the left VIM of the thalamus, and is in accord with some evidence that demonstrates thalamic involvement in lexical and semantic processes in language tasks. In the STN, we did not observe an evoked response to lexicality distinctions. Rather, we saw an overall greater low gamma power in the pseudowords trials compared to words trials, which appeared with temporal jitter from stimulus presentation to spoken response offset. And such induced gamma activity has been proposed to subserve object representation mechanisms or utilization mechanisms like updating memory contents or planning or selection of motor plans. So we think that our results support the hypothesis of thalamic selective engagement and subthalamic procedural monitoring. Thalamus is engaged in word processing at the linguistic level, possibly modulating language-relevant cortical areas through basal ganglia, thalamic cortical, and also cerebellar thalamic cortical circuits. The STN is not involved in genuinely lexical processing. Rather, we think the STN gamma activity may encode the utilization aspect of our processing, like suppression of competitors, or habitual responses, for example, a word instead of a word. For example, during reading of a word, of a non-word, FAPE, subjects have to suppress activation from real words like fate, which normally would be activated before the graphing to phoneme conversion for the pseudoword is completed. So thus we believe the role of the STN during word processing could be to contribute to the emergence of a specific behavior from competing behavioral plans. And this is all I have to share today. Thanks so much for your attention.
Yeah, thank you very much, Anna. Very exciting presentation. And thank you also for giving it at such an early time. At least I presume you're in Pittsburgh at the moment where it's quite early. And still it's so clear. Any questions, please? Right.
Great talk. Thank you so much. I wonder if you could relate the beta desynchronization findings to cortical beta desynchronization findings that have often been associated with meaning of words or meaning in general or semantics.
Yeah, that's a good point. Actually, like just looking at literature on beta oscillations, you get a lot of different kinds of betas. So there have been different interpretations of beta, what beta desynchronization means in the literature. Yes, as you are pointing out, some studies suggest that beta oscillations are involved in semantic and lexical processing. However, there are some studies that also show beta increases in response to semantic and lexical violations, for example. So based on we relate our findings to the literature on that is available on like the interpretation of the beta desynchronization in literature on movement related activities, like in the STN. This is a very characteristic, in the subthalamic nucleus, this is a very characteristic pattern that you see in response to limb movement, like with hands or fingers. And that's why it's usually taken as evidence of maintaining the status of ongoing movement while oscillations or activity at high frequency bands increases, the lower the activity at the lower frequency bands decreases. But yeah, I agree that so it could be interpreted from different angles, different ways.
Do you think, based on the circuitry, that the thalamic desynchronization drives the cortical desynchronization?
It could be, right? It could be driving the cortical desynchronization through the thalamic outflow to the cortex. But definitely there's also difference in when beta desynchronization happens in our data. In the thalamic data, it happens much earlier, like around the time when participants see the word on the screen, which could be also like pointing at the processes that are happening during that time, which is lexical access, for example, right? So yes, so maybe the two it's like patterns actually mean different things in the STN and the thalamus, and maybe the thalamic data, thalamic beta is more similar to the beta desynchronization we see in some cortical areas.
Great. I mean, often people actually see it even as preparatory before the stimulus comes on. So it'd be interesting maybe to take even a further back baseline and look to see whether it's ramping up as the stimulus is about to appear.
Well, it so like doesn't, because I showed when in our plots that I demonstrated, it is what we see in our analysis that it is time locked to the word presentation on the screen. So yeah.
Thanks. I have a related question. Do you actually know how the timing of these responses correspond to what happens at the cortical level? So which one's first in the same task?
Yeah, so we actually have cortical data. We analyze it separately, but only cortical data from the sensory motor cortex. And we see some differences in the cortex between words and pseudowords in different frequency bands at different time course.
So how is the time course? How does that time course relate to the time course in thalamus and STN?
It relates differently because there are different patterns in the cortical activity. We did not conduct, well, I mean, we did.
I'm sort of referring to Asaf's question about who drives whom.
Yeah, I'm sorry, I cannot answer that question. We tried to do coherence analysis on this data, but the results were like, we could not interpret that in a meaningful way. Yeah, I would not be able to tell with this data who drives.
Right, yeah, thank you. And another question, of course, a lot of this, as you pointed out, may be related to general motor functions.
Right.
Possibly not speech specific even. So do you have similar data on perception only, where the subjects do not have to articulate, they do not have to?
We have other data that we are currently analyzing that comes from a repetition task. So in that data, we have a listening portion where the subjects listen to syllables and then they repeat them. So we are working with that data. We're interested in also looking at what's happening during the listening portion.
Yeah.